Saturated describes an organic molecule that contains no double or triple carbon-carbon bonds, not unsaturated. Second messenger, small molecule formed in or released into the cytosol in response to an extracellular signal that helps to relay the signal to the interior of the cell. Examples include CAMP, IP3, and Ca2+. Secondary structure, regular local folding pattern of a polymeric molecule. In proteins, it refers to alpha helices and beta sheets. Secretion, production and release of a substance from a cell. Secretory vesicle, membrane-enclosed organelle in which molecules destined for secretion are stored prior to release. Sometimes called a secretory granule because darkly staining contents make the organelle visible as a small solid object. Sequence, the linear order of monomers in a large molecule. For example, amino acids in a protein or nucleotides in DNA. In general, the sequence of a macromolecule specifies its precise biological function. SER, see smooth endoplasmic reticulum. Serine threonine kinase, enzyme that phosphorylates specific proteins on serines or threonines. Sex chromosome, chromosome that may be present or absent, or present in a variable number of copies, according to the sex of the individual. In mammals, the X and Y chromosomes. Sexual reproduction, type of reproduction in which the genomes of two individuals are mixed in the formation of a new organism. Individuals produced by sexual reproduction differ from either of their parents and from each other. Beta sheet, see beta sheet. Side chain, portion of an amino acid not involved in making peptide bonds. The side chain gives each amino acid its unique properties. Signaling cascade, sequence of linked protein reactions, often including phosphorylation and dephosphorylation, that carries information within a cell, often amplifying an initial signal. Signal sequence, amino acid sequence that directs a protein to a specific location in the cell, such as the nucleus or mitochondria. Signal transduction, conversion of an impulse or stimulus from one physical or chemical form to another. In cell biology, the process by which a cell responds to an extracellular signal. Single nucleotide polymorphism, SNP, sequence in the genome that differ by a single nucleotide between one portion of the population and another. siRNA, see small interfering RNA. Sister chromatid, one copy of a chromosome, a chromatid, formed by DNA replication that is still joined at the centromere to the other copy, the pair of chromatids being known as sister chromatids. Site-directed mutagenesis, technique by which a mutation can be made at a particular site in DNA. Site-specific recombination, type of recombination that does not require extensive similarity in the two DNA sequences. Can occur between two different DNA molecules or within a single DNA molecule. Small interfering RNA, siRNA, short lengths of RNA produced from double-stranded RNA during the process of RNA interference. They base pair with identical sequences in other RNAs, leading to the inactivation or destruction of the target RNA. Small messenger, see second messenger. Small nuclear ribonucleoprotein particle. snRNP, structural unit of a spliceosome built of RNA and protein. Small nuclear RNA, snRNA, RNA molecules of around 200 nucleotides involved in RNA splicing. Smooth endoplasmic reticulum, SER, region of the endoplasmic reticulum not associated with ribosomes, involved in the synthesis of lipids. Snare, one of a family of membrane proteins responsible for the selective fusion of vesicles with a target membrane inside the cell. SNP, see single nucleotide polymorphism. snRNA, See small nuclear RNA. snRNP. See small nuclear ribonucleoprotein. Sodium pump. See Na+, K+ pump. Solute. Any molecule that is dissolved in a liquid. The liquid is called the solvent. Somatic cell. Any cell of a plant or animal other than a germ cell or germline precursor. From Greek, soma, body. Specificity. Selective affinity of one molecule for another that permits the two to bind or react, even in the presence of many unrelated molecular species. Spindle pole. One of two centrosomes in a cell undergoing mitosis. Microtubules radiating from these centrosomes form the mitotic spindle. Spliceosome. Large assembly of RNA and protein molecules that splices introns out of pre-mRNA in eukaryotic cells. Starch. Polysaccharide composed exclusively of glucose units, used as an energy store in plant cells. Stem cell. Relatively undifferentiated cell that can continue dividing indefinitely, throwing off daughter cells that undergo terminal differentiation into particular cell types. Steroid hormone. Lipophilic molecule related to cholesterol that acts as a hormone. Examples include estrogen and testosterone. Stress-gated channel. Membrane protein that allows the selective entry of specific ions into a cell and is opened by mechanical force. Stroma. 1. The connective tissue in which a glandular or other epithelium is embedded. 2. The large interior space of a chloroplast, containing enzymes that incorporate CO2 into sugars in the carbon fixation stage of photosynthesis. Substrate. The molecule on which an enzyme acts. Substratum. Solid surface to which a cell adheres. Subunit. A monomer that forms part of a larger molecule, such as an amino acid residue in a protein or a nucleotide residue in a nucleic acid. Can also refer to a complete molecule that forms part of a larger molecule. Many proteins, for example, are composed of multiple polypeptide chains, each of which is called a protein subunit. Sugar. A substance made of carbon, hydrogen, and oxygen with the general formula CH2O(N). A carbohydrate or saccharide. The sugar of everyday usage is sucrose, a specific sweet-tasting disaccharide produced by beet or sugarcane. Sulfhydryl group, SH-thiol. Chemical group containing sulfur and hydrogen bound in the amino acid cysteine and other molecules. Two sulfohydryls can join to produce a disulfide bond. Survival factor. Extracellular signaling molecule that must be present to prevent apoptosis. Symbiosis. Intimate association between two organisms of different species from which both derive a long-term selective advantage. Synapse. Specialized junction between a nerve cell and another cell. Nerve cell, muscle cell, gland cell, across which the nerve impulse is transferred. In most synapses, the signal is carried by a neurotransmitter, which is secreted by the nerve cell and diffuses to the target cell. Synaptic vesicle. Small membrane enclosed sac filled with neurotransmitter that releases its contents by exocytosis at a synapse. Telomerase. Enzyme that elongates telomeres, the repetitive nucleotide sequences found at the ends of eukaryotic chromosomes. Telomere. Structure at the ends of linear chromosomes, associated with a characteristic DNA sequence that is replicated in a special way. Counteracts the tendency of the chromosome otherwise to shorten with each round of replication. From Greek, telos, end. Telophase. Final stage of mitosis in which the two sets of separated chromosomes decondense and become enclosed by nuclear envelopes. Template. A molecular structure that serves as a pattern for the production of other molecules. Thus, a specific sequence of nucleotides in DNA can act as a template to direct the synthesis of a new strand of complementary DNA. Tight junction. cell-cell junction that seals adjacent epithelial cells together, preventing the passage of most dissolved molecules from one side of the epithelial sheet to the other. Tissue, organized mass of cells with a specific function that forms a distinctive part of a plant or animal. Thioester bond, high-energy bond formed by a condensation reaction between an acid, acyl group, and a thiol group, SH, seen, for example, in acetyl-CoA and in many enzyme substrate complexes. Trans, beyond or on the other side. Transcription, a process that uses one strand of DNA as a template to synthesize a complementary RNA sequence, sometimes termed the primary transcript, catalyzed by the enzyme RNA polymerase. Transcription factor, term loosely applied to any protein required to initiate or regulate transcription in eukaryotes, includes transcription regulators as well as the general transcription factors. Transcription regulator, protein that binds specifically to a regulatory DNA sequence and is involved in controlling whether a gene is switched on or off. Transfer RNA, tRNA, set of small RNA molecules used in protein synthesis as an interface, adapter between mRNA and amino acids. Each type of tRNA molecule is covalently linked to a particular amino acid. Transformation, process by which cells take up DNA molecules from their surroundings and then express genes on that DNA. Transgenic organism, a plant or animal that has stably incorporated one or more genes from another cell or organism and can pass them on to successive generations. Trans-Golgi Network, TGN, that part of the Golgi apparatus that is furthest from the endoplasmic reticulum and from which proteins and lipids leave for lysosomes, secretory vesicles, or the cell surface. Transition state, chemical structure that forms transiently in the course of a reaction and has the highest free energy of any reaction intermediate. Translation, process by which the sequence of nucleotides in a messenger RNA molecule directs the incorporation of amino acids into protein, occurs on a ribosome. Translation initiation factor, protein that promotes the proper association of ribosomes with mRNA and is required for the initiation of protein synthesis. Transport vesicle, membrane vesicles that carry proteins from one intracellular compartment to another, for example from the ER to the Golgi apparatus. Transporter, membrane protein that transports ions or molecules across a cell membrane. Transposon, general name for short segments of DNA that can move from one location to another in the genome, also known as mobile genetic element. Triacylglycerol, glycerol ester of fatty acids, the main constituent of fat droplets in animal tissues, where the fatty acids are saturated, and of vegetable oil, where the fatty acids are mainly unsaturated. tRNA, see transfer RNA. Tryptophan repressor, a bacterial protein that, in the presence of tryptophan, binds to a specific region of DNA and shuts off production of the tryptophan biosynthetic enzymes. Tubulin, protein from which microtubules are made. Tubulin ring, protein complex in centrosomes that nucleates microtubule assembly. Tumor suppressor gene, a gene that in a normal tissue cell inhibits progress through the cell cycle. Loss or inactivation of both copies of such a gene from a diploid cell can cause it to divide as a cancer cell. Turnover number. In enzyme catalysis, the number of substrate molecules processed to product per second per enzyme molecule. Although different types of enzymes can have very different turnover numbers of 1,000 or more are quite common, a reflection of the impressive catalytic power of enzymes. Tyrosine kinase, enzyme that phosphorylates specific proteins on tyrosines. Unfolded protein response, Cellular response triggered by the accumulation of misfolded proteins in the endoplasmic reticulum. The cell produces more endoplasmic reticulum and more of the molecular machinery needed to restore proper protein folding and processing. Unsaturated describes a molecule that contains one or more double or triple carbon-carbon bonds. Vmax, the maximum rate of enzymatic reaction, attained immediately after the addition of substrate at a concentration sufficient to fully occupy the active sites of all enzyme molecules present. Valence, for an atom, the number of electrons that it must either gain or lose, whether by electron sharing or by electron transfer, to achieve a filled outer shell most readily. Thus, for example, the valence of Na is 1, it must lose 1 electron, and the valence of Cl is 1, it must gain 1 electron. The valence of an atom is equal to the number of single bonds that the atom can form. Van der Waals force, attractive force due to fluctuating electrical charges that come into play between two atoms that are 0.3 to 0.4 nanometers apart. At a shorter distance, repulsive forces begin to operate. Vector, genetic element, usually a bacteriophage or plasmid, that is used to carry a fragment of DNA into a recipient cell for the purpose of gene cloning. Vesicle, small, membrane-enclosed, spherical organelle in the cytoplasm of a eukaryotic cell. Vesicular transport, transport of material between organelles in the eukaryotic cell via membrane-enclosed vesicles. Virus, particle consisting of nucleic acid, RNA or DNA, enclosed in a protein coat and capable of replicating within a host cell and spreading from cell to cell, often the cause of disease. Voltage-gated channel, membrane protein that selectively allows ions such as Na+, carried by the voltage-gated Na+ channel, to cross a membrane and is opened by changes in membrane potential, found mainly in electrically excitable cells such as nerve and muscle. Wild type, normal, non-mutant form of a species resulting from breeding under natural conditions. WNT protein, member of the WNT family of extracellular signaling proteins with many roles in development, including the maintenance of stem cells in a proliferative state. X chromosome, one of the two sex chromosomes in mammals. The cells of men possess one X and one Y chromosome. X-ray crystallography, technique used to determine three-dimensional protein structures by analyzing the diffraction pattern of a beam of X-rays passed through a crystal of the protein. Y chromosome, one of the two sex chromosomes of mammals. The cells of women contain two X chromosomes. Yeast, common term for several families of unicellular fungi. Eukaryotic cells used as model organisms, include species used for brewing beer and making bread, as well as species that cause disease. Zygote, diploid cell produced by fusion of a male and a female gamete, a fertilized egg.